This video is going to talk about muscle tissue histology and the neuromuscular junction. I want to draw your attention to the left side of the screen where we see the model of the neuron. On the left side of the model, you will see the cell body of the neuron. The cell body of the neuron is located near the spinal cord, and it contains the biosynthetic center of the cell. What we mean by biosynthetic center is that it contains all of the organelles that are necessary to sustain the function of the cell. You will see that extending from the biosynthetic, the cell body, is the axon, and the axon meets the cell body at a structure called the axon hillock. So if the cell body is located near the spinal cord, and this neuron is going to attach to skeletal muscle, the axon could be as long as your arm or your leg. The axon will continue until it terminates at a structure called the axon terminal. The axons will branch into many axon terminus. The next model that you can see here is an enhanced magnification of the axon terminal. At the very top of this model would continue the axon up toward the cell body. What we can see at the bottom of the model is that the axon terminal widens to almost look like a foot process. We can see lots of structures inside the axon terminal. We can see red mitochondria. And we can see lots of little bubbles at the very terminal part of the axon terminal. These little bubbles are called synaptic vesicles, and they are filled with a neurotransmitter. The neurotransmitter is the chemical that allows the neuron to communicate with another type of tissue. The synaptic vesicles allow the neuron to communicate with other cells. The synaptic vesicles are going to undergo exocytosis to release their neurotransmitter to the extracellular space. The clear plastic in this model is supposed to illustrate the muscle tissue. There's a small gap between the axon terminal and the muscle tissue that we would call the synaptic cleft. So those synaptic vesicles contain a neurotransmitter called acetylcholine. Acetylcholine is going to stimulate the muscle tissue to contract. You'll learn more about the mechanism for that in lecture. I want to draw your attention to the right side of your screen to the tissue slide that we have on the microscope. This is the neuromuscular junction. So in very dark, we can see the axon, and we can see that it's a really large bundle that will branch. Those branches are going to end with multiple branching axon terminals. We're going to zoom in from the 10x objective to the 40x objective and take a closer look at the axon terminals. So here we can see the branching axon that ends in these bubble-like structures called axon terminals. So this slide illustrates the junction between muscle tissue, which you'll see in pink, skeletal muscle tissue, and the black is the neuron, and the neuromuscular junction is the junction between the neuron and muscle tissue.